Okay, so welcome to this next video on endothelium-derived relaxation factor. Okay, so in the previous video what we saw is that if we put acetylcholine onto an endothelial cell, it will activate M3 muscarinic acetylcholine receptors, which will cause the activation of the alpha-Q GTP subunit. So the alpha-Q subunit here has now bound GTP basically. So this is alpha Q and it's bound to GTP, guanosine triphosphate. So G proteins always have an on and an off state. In the on state they're bound to GTP, and in the off state they're bound to GDP. So this alpha Q subunit is now in the on state since it's bound to GTP. Okay, now what this alpha-Q GTP subunit is going to do is it's going to activate an enzyme in the phospholipid bilayer of cells. And that enzyme is an enzyme known as phospholipase C, and it's specifically of the beta type. So let me show this in the membrane. So here's the phospholipid bilayer here. And this enzyme, its full name is phospholipase C, and it's of the beta type. Phospholipase C of the beta type. Right, and now what's the function of this enzyme? So I'll just firstly colour this enzyme in some nice purple colour. Okay, and now what we want to see is what the function of this enzyme is. So basically, when alpha-Q-GTP comes along and binds to the phospholipase C-beta subunit, it's going to be activated. So what does it do? Well, basically, it's going to break down a usual component of the phospholipid bilayer. And this usual component of the phospholipid bilayer is a PIP2 molecule. So let me show you the structure of a PIP2 molecule. So basically, it's based on the normal structure of a phospholipid. So let me show you the structure of a normal phospholipid. So basically, a normal phospholipid has these two long-chain carboxylic acids which are esterified to two hydroxyl groups of glycerol, and which are called the hydrophobic tails. So let me colour these in. So let's colour them in green. Okay, so in light green we have these long-chain carboxylic acids, which are also called fatty acids. So these are fatty acids which are esterified to the first and the second hydroxyl groups of the glycerol molecule. Okay, and they are the hydrophobic tails of the phospholipid molecule. Then in orange, and represented by this horizontal line, we then have the glycerol molecule itself. So this is the backbone, really, for all phospholipids. So this is glycerol. Okay, or if you're being a chemist, propane 1,2,3-triol. So let me just write that down. So its proper name is propane 1,2,3-triol, although glycerol is still extremely pervasive among biochemists. Okay, and then off the third hydroxyl group of the glycerol molecule, you then have a phosphate group bound. So you have a phosphate group here. Okay, so this is a phosphate group bound off the third hydroxyl group of the glycerol molecule. Right, so that's the structure of a normal phospholipid. Now, another name for a phospholipid is a phosphatidate molecule. So often you will obviously call phospholipids phospholipids, but the old name, I think, for a phospholipid is that it's a phosphatidate molecule. Is this going to fit in? Phosphatidate, there we go. Phosphatidate molecule. Okay, and this now allows us to get some sort of insight into what PIP2 is. Because if we look at the full name for PIP2, so here's PIP2. So this is the molecule which phospholipase C enzymes are going to break down. And what it stands for, in full, is phosphatidyl. Phosphatidyl. So that's the P at the front. Inositol, that's the next I in its name. And then 4,5-bisphosphate, so that's the P2 on the end, to denote that you have two phosphate groups sticking off the end of this. Okay, right. So this phosphatidyl name right at the beginning here, so this bit here if this is a phosphatidate molecule, then that suggests that it has this structure coming off it as a group. So it's inositol with a phosphatidate molecule sticking off the side of it. And indeed, that intuition would be correct. Basically, what you do is you stick an inositol molecule off this phosphatidate molecule, and that creates you phosphatidyl inositol. So it's named kind of the opposite way round. It's named as though you start with the inositol and you've stuck the phosphatidate onto it. So it's phosphatidyl group stuck off inositol. And the way you stick the inositol onto the phosphatidate molecule is you stick it onto the other hydroxyl group at the phosphate group, basically. Okay, so this 6-carbon ring here is inositol. So inositol is a 6-carbon ring where all of the bonds in the 6-carbon ring are single bonds, and off all of the carbons you also have a hydroxyl group. So one hydroxyl group comes off every single one of these 6 carbons, and you also then have a hydrogen making up the final bond. So it's a 6-membered carbon ring with hydroxyl groups and hydrogens off every single one of the 6 carbons. Now, basically, we have formed a condensation, we've undertaken a condensation reaction between the hydroxyl group off this carbon of the inositol ring and the hydroxyl group that's free on this phosphate group. And that's how we have created phosphatidyl inositol. So in blue here, that's the inositol here, okay? Right, and the phosphatidyl, I will circle that. That's the entire of this structure here, basically. Okay, right. Now, we're not finished. That's the structure of phosphatidyl inositol. To turn it into a phosphatidyl inositol 4,5-bisphosphate, we need to add 2 phosphate groups on, one on the 4th and one on the 5th carbon. So, the way the carbons in the inositol molecule are labelled is this one's labelled as 1, this one 2, 3, 4, so that is the 4th phosphate, 5, and then 6. Okay, so we've got phosphatidyl inositol 4,5-bisphosphate. Okay, so these are phosphate groups stuck off the edge. So that is now a complete PIP2 molecule. And for the life of me, I do not know why it's not called phosphatidyl inositol 3,4-bisphosphate. Someone decided long ago that the carbon with the phosphate group here should be the 5th carbon in the inositol ring. Why they didn't think that should be the 3rd carbon, I don't know, but we're stuck with it. Right, so what's phospholipase C going to do to this, this molecule, which we can see now is just a modified phospholipid. It's just a phospholipid with this structure stuck on the end of it, basically. And it's a usual component of the phospholipid bilayer. You do have this in your phospholipid bilayers. Okay, right. So what's phospholipase C going to do? Well, basically, it's going to cleave a bond in this PIP2 molecule and break it down into 2 components. It's specifically going to cleave this bond here between the glycerol and this phosphate group. So, let's have a look at the two structures we're going to get out of that cleavage. We are going to get this structure here, which is glycerol with these 2 fatty acids still esterified to it. So here's glycerol in orange, and here in green are the long-chain carboxylic acids, or the fatty acids, which are esterified to the first 2 hydroxyl groups of the glycerol molecule. Now, this structure is known as a diacyl glyceride because you have 2 acyl groups bound by ester links to a glycerol molecule, and in short it's denoted DAG, so diacyl glyceride. Okay, the other structure we're going to get off is this inositol ring here. This is 6-membered carbon ring that is inositol with these 3 phosphate groups stuck off the side, so 1, 2, and then 3 here. Okay, so let's circle, we'll colour in this one as well. So here's blue for inositol, there, and then we've got these 3 phosphate groups coming off of it. Okay, 1 here, 1 here, and 1 here. Now, that molecule is known as inositol because you've got the inositol ring, and then you've got these 3 phosphate groups coming off it. So it's inositol 1, 4, 5 tris phosphate. Okay, and it's often just called inositol trisphosphate, so you omit the numbers, or even more commonly it's called I for inositol, and then P for phosphate, and then 3 to denote that you have 3 phosphate groups. So it's very common to see that called IP3. Okay, now both of these lead to downstream signaling pathways within the endothelial cell. Now, we're not interested in what diacylglyceride is going to do. It's going to go off and activate protein kinase C, which is very important. But for our purposes, inositol 1,4,5-trisphosphate is going to be the important molecule as far as we're concerned. Okay, so now what we need to discuss is the fact that IP3 will be going up in the entire endothelial cell. So if I draw the endothelial cell back again, so here's our endothelial cell here. Now, most of the endothelial cell is exposed basically to the blood basically. So this is the blood here. Now, if I've given you an intravenous dose of acetylcholine, then all of your M3 receptors basically will be being activated by acetylcholine. So all over the cell surface, you are going to get the activation of these M3 receptors by acetylcholine, which I'll just denote as ACH. So over here as well, you're going to get acetylcholine binding and activating these M3 receptors here. Okay, now, downstream of the M3 receptor activation is the production of IP3. So all over the cell, you're going to get production of IP3, inositol 1,4,5-trisphosphate. So IP3 level is going to go up in the entire cell, and that's going to be important for the type of signaling we get. It's basically going to be a global signal, basically. IP3 is going up everywhere. It's not just going up in some specific little region of the cell, it's going up everywhere. Okay, and I'll continue this story in the next video.